Humans have long held a fascination for the human brain. We chart it, we've described it, we've drawn it, we've mapped it. Now, just like the physical maps of our world that have been highly influenced by technology, think Google Maps, think GPS, the same thing is happening for brain mapping, true transformation. So let's take a look at the brain. Most people, when they first look at a fresh human brain, they say it doesn't look like what you're typically looking at when someone shows you a brain. Typically what you're looking at is a fixed brain. It's gray. And this outer layer, this is the vasculature, which is incredible around the human brain. This is the blood vessels. 20% of the oxygen coming from your lungs, 20% of the blood pumped from your heart is servicing this one organ. That's basically, if you hold two fists together, it's just slightly larger than the two fists. Scientists, sort of in the end of the 20th century, learned that they could track blood flow to map non-invasively where activity was going on in the human brain. So, for example, they can see in the back part of the brain, which is just turning around there, there's the cerebellum. That's keeping you upright right now. It's keeping me standing. It's involved in coordinated movement. On the side here, this is the temporal cortex. This is the area where primary auditory processing, so you're hearing my words, you're sending it up into higher language processing centers. Towards the front of the brain is the place in which all of the sort of more complex thought, decision-making is the last to mature, sort of late adulthood. This is where all your decision-making processes are going on. It's the place where you're deciding right now you probably aren't going to order the steak for dinner. So if you take a deeper look at the brain, one of the things, if you look at it in cross-section, what you can see is that you can't really see a whole lot of structure there. But there's actually a lot of structure there. It's cells and it's wires all wired together. So about 100 years ago, some scientists invented a stain that would stain cells. And that's shown here. In the very light blue, you can see areas where neuronal cell bodies are being stained. And what you can see is it's very non-uniform. You see a lot more structure there. So the outer part of that brain is the neocortex. It's one sort of continuous processing unit, if you will. But you can also see things underneath there as well. And all of these blank areas are the areas in which the wires are running through. They're probably less cell dense. So there's about 86 billion neurons in our brain. And as you can see, they're very non-uniformly distributed. And how they're distributed really contributes to their underlying function. And of course, as I mentioned before, since we can now start to map brain function, we can start to tie these into the individual cells. So let's take a deeper look. Let's look at neurons. So as I mentioned, there are 86 billion neurons. There are also these smaller cells, as you'll see. These are support cells, astrocytes, glia. And the nerves themselves are the ones who are receiving input. They're storing it. They're processing it. Each neuron is connected via synapses to up to 10,000 other neurons in your brain. And each neuron itself is largely unique. The unique character of both individual neurons and neurons within a collection of the brain are driven by fundamental properties of their underlying biochemistry. These are proteins. They're proteins that are controlling things like ion channel movement. They're controlling who nervous system cells partner up with. And they're controlling basically everything that the nervous system has to do. So if we zoom in to even deeper level, all of those proteins are encoded by our genome.